revision class from microbiology with Shagnik. So it is another aspect of methods in molecular cloning, that is electrophoresis. Now you may ask why I am telling that it is a part of methods in molecular cloning. So the fact is, you have cloned your DNA well. If you want expression, then you also perform translation inside the host cell too. Then now suppose if you want to find out your gene of interest from the recombinant clones, right? Because your recombinant colonies will be in the petri plate in the form of colony if you have used plasmid, and in the form of flux if you have used fudgemid, right? Now how can you find out your gene of interest containing colonies? Okay, so for such kind of screening purpose, you need a procedure that I have already discussed with you in our earlier videos, that is the southern blotting technique, right? And for southern blotting and for western blot and northern blotting, we need agarose gel electrophoresis. That is the reason why our today's topic of revision from recombinant DNA technology is the electrophoresis. Although I should say that it is also a revision class of bioinstrumentation too, right? Although I think I have not discussed about any video regarding gel electrophoresis or agarose gel. We have discussed about SGS page. Okay, so let's start. So let's start with a basic introduction of electrophoresis. So electrophoresis is the combination of two words. One is electro, another one is phoros. Phoros is the Greek verb which means carry across. That means here we are using electrical energy to move charged particle through an electrical field, and with the help of this phenomenon we are simply separating our molecules from a complex mixture. So electrophoresis can be of two types: gel electrophoresis, 2D gel electrophoresis. Gel electrophoresis, depending on the gel used, can be divided into two types: agarose gel electrophoresis, polyacrylamide gel electrophoresis. Agarose gel can be used for DNA RNA, and polyacrylamide gel is used for protein and low molecular weight DNA and low molecular weight RNA. So we can also divide polyacrylamide into two kind of polyacrylamide gel. One is native phase, where we are using where we do not use any kind of reducing agent, okay, and another one is SGS phase. So that is the general classification of gel electrophoresis. Now the question is, why DNA RNA in case of agarose and protein, and low molecular weight DNA and low molecular weight RNA will be in case of polyacrylamide? So the answer lies in another question, that why we need agarose and polyacrylamide gels. Because agarose and polyacrylamide gels together cross-link with each other and forms a mesh-like structure, and in the mesh-like structure there are various kind of pores, okay, and with the help of this pore we are separating our biological samples, or I should say charged molecules, according to their molecular weight mainly, okay. So in case of agarose, the pore size generated by agarose is relatively higher, okay. That means in case of agarose, pore size that I will mention in form of A in suffix P is greater than the polyacrylamide. That is why protein, low molecular weight DNA and low molecular weight RNA can be separated with the help of polyacrylamide, and intact DNA and RNA can be separated with the help of agarose, okay. So it is a total funda of pore size. Now let's move to another kind of gel electrophoresis, that is 2D gel electrophoresis. So why, remember one thing, that 2D gel electrophoresis is only used for protein sample separation. Now my question is, why we use 2D gel electrophoresis, and what is 2D gel electrophoresis? So 2D gel electrophoresis is simply protein separation process in which we take two different characteristics under consideration to separate our protein sample. One is isoelectric focusing, another one is molecular weight. So now the question is why we need two different properties to separate the protein molecules. Because remember, in a complex protein sample there may be a possibility that there are multiple protein component present of same molecular weight. Suppose your protein of interest belongs among them, then how can you separate your protein of interest from those bunch of protein samples of same molecular weight? So in that kind of situation you have to need another characteristics that will separate your protein of interest from those protein of interests with the help of 2D gel electrophoresis. Okay, so that is the reason in case of 2D gel electrophoresis we take two properties under consideration. So let's discuss about the components of agarose in a little bit brief. So agarose has higher pore size than polyacrylamide, I have discussed with you. Now the question is from where agarose is derived. So agarose is coming from agar. If I zoom this picture, then it will be very easy to you to follow. So agar is the main component which we all know that used as a solidifying agent, right? It is consist of two components, agarose and agaropectin. Agarose is the linear polymer repeating monomeric unit and it is the predominant part. Okay, and this agarose is in turn consists of agarobiose. This agarobiose is repeating monomeric unit. Sorry, I have made a mistake that I have told you that agarose is the repeating monomeric unit, but it is not. Agarobiose is the repeating monomeric unit. Okay, and agarose is the predominant part, and this repeating monomeric unit agarobiose together joins and forms agarose. Okay, and this agarobiose in turn formed by two components, one is D-galactose, another one is 3,6-anhydro L-galactopyranose. Okay, so that is an agaropectin is the heterogeneous mixture of smaller molecules that occur in less amount. So agar is divided, it is consist of agarose and agarobiose. Agarose is in turn consist of agarobiose. Sorry, agar is consist of agarose and agaropectin. Okay, and agarose is in turn consist of agarobiose, and this agarobiose is in turn consist of D-galactose and 3,6-anhydro L-galactopyranose. So that is the idea. Okay. Now let's move to the polyacrylamide part. Polyacrylamide consists of two things. One is acrylamide, another one is N-prime methylene bisacrylamide. These two components together join cross-linked with the help of APS and TEMED. APS means ammonium persulfate and TEMED together helps in cross-linking of acrylamide and N-prime methylene bisacrylamide and gives us polyacrylamide. This polyacrylamide is more costlier than that of agarose. You need to remember that is why in case of normal separation we prefer agarose. But if we are dealing with two smaller molecules, then we have to move for polyacrylamide. Okay. So let's enter into our today's topic, that is agarose gel electrophoresis. So nucleic acid separation technique. So how can you separate agarose nucleic acid with the help of agarose gel electrophoresis? Despite discussing that diagram, I will later give you that note so that you can take a screenshot. Let's move to the diagram first so that it will be very easy to you to remember. This is the diagram. This diagram I have taken from Microbe Notes. If you want to download this picture, you can simply go to Google and search this, and you can easily find out the picture. Okay. So our first target will be the preparation of agarose gel. Although this picture is not completed yet, because we have to prepare first agarose gel not in the casting tray but in a conical. Okay, we can also prepare our agarose gel in a casting tray too, that not a big deal. So how can we prepare agarose in the conical? So there is a, they can tell you either to prepare 1.5% agarose gel or to prepare 0.8% agarose gel. You have to prepare. And how, why they will mention you the agarose concentration? Because remember, the concentration is in directly proportional to the pore size. If you use 1.5% agarose, then your pore size will be I have again made a mistake that concentration will be inversely proportional to the pore size. If 1.5% agarose gel you use, your pore size will be smaller. If you use 0.8% agarose, your pore size will be higher. Okay, the explanation is very simple, that you are used a higher concentration agarose, that means there are more agarose, so they will more densely cross link. That is why the pore size generated by them will be small than that of 0.8%, and it is totally done on the basis of our sample of interest. If our sample of interest is of lower molecular weight, then we will prefer 1.5% agarose. If our sample of interest is of higher molecular weight, then we will prepare 0.8% agarose. So how can we prepare 1.5% agarose? 1.5% agarose is prepared with the help of 1.5 gram agarose along with 100 milliliter buffer. This buffer is usually tris acetate EDTA buffer or tris borate EDTA buffer. Okay, let's discuss about TAE. So tris acetate is used to maintain the pH 8, and EDTA is acting as a chelating ion and chelates divalent cation. Now the question is, what is the advantage of chelating divalent cation? Remember, these Mg2 plus ions or other divalent cations play a role of cofactor of DNase enzymes and RNase enzymes. If you are willing to separate RNA, then you have to keep your RNA safe from RNase, and if you are willing to separate DNA, then you have to keep your DNA from DNase safe, right? So that is why if you chelate the Mg2 plus ion with the help of EDTA, so the DNA will not activate because it lacks its cofactor, and we all know that enzyme substrate reaction cannot occur with the help of cofactors, right? So that is the reason DNase and RNase, if present in the medium, will become inactivated and our DNA sample will not be destroyed. That is the utility of using EDTA. So agarose composition is 1.5% agarose, 1.5 gram agarose, 100 microliter buffer. It can be TAE or TBE depending on our resources, and but remember one thing, TAE is more preferable than TBE because TBE is involved in some undesirable reaction with DNA. That is why we usually prefer TAE. But if you do not have TAE, then you have to move for TBE. You do not have any kind of another options. So now we will once our gel is solidified, we have simply remove our comb so that well will be generated, and as a result this well we can use to put our samples, right? Now we have to prepare our DNA samples. How can we prepare the DNA samples? So here we have to add two things. One is our tracking dye. Tracking dye can be bromophenol blue or xylene cyanol. Okay, and glycerol can be used so that it will increase the density of the sample. Okay, and as the density of the sample is increased, so that it can be easily layered into the top into the bottom of the well, so that it cannot so that it can be easily loaded inside the well. Suppose, let me give you a picture so that it can be easy to interpret. Let's see that this picture. So we have added the glycerol so that our gel can be easily layered inside the well, okay, because its density is increased by the glycerol. Because remember one thing, that DNA RNA are relatively lighter in weight, so if you do not add glycerol, the DNA and RNA molecule cannot stable inside the well. They will just remove from the well and either mix with the gel or mix with the buffer. That is why we have to settle the DNA molecule inside the well. That is why we have to add glycerol. Okay, so glycerol, and what is the utility of using tracking dye, that is xylene cyanol and bromophenol blue? Let me discuss. Okay, I can also discuss with you in that picture too. So suppose that you have started the gel electrophoresis without adding tracking dye. What will happen, as DNA is usually don't have any kind of color, right? So you cannot track your DNA running during the gel electrophoresis live. So what is the disadvantage of not able to track them? So that your DNA will be crossed, DNA will cross the gel and it comes in the buffer and eventually loss. Okay, and you cannot prevent the loss unless or until you can track them how much they run, right? That is the reason tracking dye is required to track them live during gel electrophoresis. Okay, now before loading our DNA sample inside the gel, we have to first insert our gel casting tray. Suppose this green box is a gel casting tray in a gel tank. This gel tank is filled with the buffer TBE or TAE. Okay, so why we have put our casting tray inside a gel tank? So the answer is very simple, that so that our gel cannot dry. Okay, we have to keep in it our mind, because otherwise our gel will become dry and it is not required. So that is the reason why we have to put our gel before loading our DNA sample inside a gel tank. Okay, so now we will add all the electrodes, that is positive electrode and negative electrode, at appropriate direction. That is important. The negative electrode's direction will be towards the well. That means here will be the negative electrode and positive electrode will be here. Why? Because we all know that DNA is negatively charged due to the presence of phosphodiester backbone, right? So that is the reason we have to put our DNA, we have to set negative electrode at that direction and positive electrode at that direction. Okay, so we will load our sample then. So sorry, we have already loaded our sample, then we have added our positive electrode, negative electrode, and then we will simply switch on. So our voltage will be here 80 to 150 volt for 45 to near about 1 minute, 1 hour, depending on the sample we are willing to separate. Okay, so well, so we have separated our DNA on the basis of their molecular weight. So now the idea will be, we have completed the electrophoresis, so now we have to visualize our separated DNA band, right? So how can we separate? Remember, after the perfect dissolution of agarose and chilling of them, we have added ETBR in that solution. Okay, so ETBR is a intercalating agent and it can be act as a fluorescence molecule in presence of UV ray. Okay, so with the help of this ETBR, we will simply transfer our gel inside a gel dock. Okay, this gel dock is something like that. Okay, or in a UV trans illuminator. So in presence of the UV light, our ETBR will fluoresce and they will help the DNA molecule to visualize, as they are also intercalating agent and intercalate with the DNA molecule. Okay, so that is how we can visualize our DNA band with the help of UV illuminator and biorad machine, that is gel dock. Now as we have added the marker DNA in the first well, I have mentioned about marker, I cannot remember it, so whatever. Remember one thing, that in the first well we have to add marker DNA, which is of known molecular weight. This marker DNA is also often known as ladder DNA and stuff. So this marker DNA or ladder DNA will guide you to determine your the molecular weight of your DNA molecule or RNA molecule. This ladder DNA can be of various size. If it is of 1 kb, then make sure that your band will be something like that. That means the smaller one will be 1 kb, the higher than the smaller one will be 2, 3, 4, 5, 6. In that way the band will be arranged, and on the basis of those band you can interpret your the molecular weight of your band DNA. That is how the Agarose gel electrophoresis technique is performed. Okay, so this is the DNA separation method, and here question will come from various aspects. First of all, they can ask you why ETBR is needed. It is an intercalating agent and help us to visualize the DNA bands. Okay, they can also ask what can you do to dissolve agarose. We have to microwave our sample for near about 2 minutes to dissolve agarose. Okay, they can also ask what is the utility of xylene cylon and bromophenol blue. So they are tracking dye and they simply track our DNA molecule live during the gel electrophoresis. Another important question will be what will happen if you set your electrodes in wrong direction. That means positive electrode in the direction of well and negative electrode in the opposite direction. So in such cases the direction of your DNA sample will be in that way, and your DNA will be eventually lost in the buffer. Okay, it is another kind of important question. So they will also give you a kind of analytical question, that suppose you are a research scholar and your guide mentioned you to prepare 1.5% agarose and 0.8% agarose gel both for other research scholar, and now two of them will ask you to give them a agarose solution so that they can separate 1 kb DNA and 500 base pair DNA. So what solution will you prefer among 1.5 and 0.8%, to whom and why? So I will prefer 1.5% for the person who wanting to separate 500 base pair DNA, because 500 base pair is relatively smaller than the 1 kb. 1 kb is relatively larger, and I will prefer him or her to use 0.8% agarose gel solution to separate 1 kb DNA, because 1 kb is relatively larger than the 500, and I have told you that all is the matter of porosity, pore size. Okay, so this is one kind of question. So that's is a different kind of question that they can ask. So we have today all again revised another important topic of methods in molecular cloning. So I will hold the these slides so that you can take a screenshot. So okay, so I will again come back with another topic from methods in molecular cloning for our revision purpose, and in that topic I will start discussion about Agarobacterium tumefaciens mediated gene transfer. That is the last gene transfer that we have. Then we will discuss about southern blot, and eventually our chapter will be finished. So till then, bye bye.